Welcome and thank you so much for joining us at today's open session of our All of Us Research Program Advisory Panel. I'm Rachel Peterson, the Chief of Staff for the All of Us Research Program, and also I myself am a participant. First, I want to give a couple of housekeeping notes to share. As attendees, you'll note that the video and audio are turned off. You may submit questions for our Q&A session through the private Q&A box, or if you have any technical issues. And depending on time, we may respond privately, individually to questions. But due to high attendance and volume, we may not be able to address or respond to every question during the session. To follow up, if you choose, you can share your email address, and then we can work to respond to you as well. But we will be answering some of the questions we received today at the end of the session. But with that, I would like to turn it over to our CEO, Josh Denny of the All of Us Research Program. Josh, please take it away.
Thank you so much, Rachel. It is a pleasure to have all of you with us today, and I appreciate you taking your time to join us for this advisory panel open session. I am going to give you a sense of the current progress of the program and some of what we had planned for the future. First, I want to thank all the advisory panel members that are with us today, pictured here, and especially a warm welcome to our two new members on the bottom right, Adriana Mares and Michelle Anderson, who will be representing the participant and the researcher perspective. These advisory panel has been so helpful for us as a working group of the Council of Councils, which was actually last week, to help us think of our strategic vision, provide us important insight, and represent just a number of different domains of thought and experiences to help guide us to the greatest impact we can have. Last year, this working group produced a strategic report which looked at where we are and where we should be going in light of budgetary changes and other challenges, as well as important opportunities in front of us. And I just want to reflect a minute. It's been 10 years since Congress passed the very bipartisan 21st Century Cures Act, which formally established the All of Us Research Program, then called the Precision Medicine Initiative Cohort Program, as a part of an enduring legacy to make an impact on precision medicine for all populations in this country and really accelerate the pace in which we can do science with a U.S. national program. And really, I would say All of Us has been the most enduring manifestation of this vision. And on the right, you see photos from the 6 different cities in which we did launch events across the country on May 6, 2018. Very rapidly getting us started. And this just gives you a sense of the timeline of the program. We have become one of the largest research cohorts in the world and represent all parts of the United States and really all communities, geographies, experiences, lifestyles, and environmental exposures and genetics that is part of this rich country. And you can see our pace here. We launched nationally in 2018 after a rapid buildup of the program. We launched to researchers in 2020 during COVID, where researchers could start accessing the database. And we set a 5-year goal to, by the end of 2026, so still ahead of us, to try to get to 10,000 researchers, which was actually an ambitious goal. But you can see that we reached 10,000 researchers in 2024. We are now over 23,000 researchers using the program. And they're really discovering important findings that are, we'll talk about some of these that are really translating. We started our pediatric pilot in 2024, and we now have over 3,000 children that are part of the program. And importantly, over 880,000 participants that have joined us. And I thank all the participants that are with us today and in this meeting and so many others. We often say that, and it's a quote that has always stuck with me, that science moves at the speed of trust. And we could not represent the entire United States and 98% of all 3 zip codes if we did not have such a strong arm of engagement and partners across the country. We have engagement partners that represent national engagement partners, that represent local. We have healthcare provider organizations across the country that have really importantly partnered with us to convey trust in this truly national program. These individuals volunteer to share all sorts of information. More than 90% say that we can have electronic health records as really important longitudinal records of the diseases and health trajectories that people experience. Collecting those data in the United States is much harder. We have about 500,000 for which we've collected those data, but we continue to make tremendous progress and pioneer new ways in which those data are collected and aggregated into harmonized data models to make them useful for research across many different healthcare systems and many different EHR vendor systems, and allowing people for whom we don't have that can enroll from anywhere to collect and donate those information. Importantly, individuals have donated biosamples as well. That includes DNA, RNA, serum plasma, and urine as part of what we have, and those really enable discovery. Now, one of the things we wanted to do differently in this program was return information to participants. We heard as we were developing this program that it was really key that we could translate this, and people wanted to learn about precision medicine. They wanted to learn about things that might affect their healthcare. And so one of the most important things that we've done with that is returning health-related DNA results. And on the left panel here, you can see that we have returned health-related DNA results to over 200,000 participants. And the top line here gives hereditary disease risk. So about 2.8% overall of individuals had risk of a genetic change that gave risk to cancers, cardiovascular disease, and other conditions for which there are interventions available. And so these are, potentially dramatic, potentially life-saving events. I'll give you some examples later. You can see, though, that the percentage of people that we found actionable genetic variants for actually does vary by self-identified race and ethnicity. And some of this probably represents our ability to accurately interpret genetic variants across different populations. So this is a really important reason why this program exists, is without collecting data from diverse populations of diverse genetic ancestries, we have a harder time interpreting genetics for everyone. And many studies have shown this. Importantly, on pharmacogenetics, on the bottom row, it are genetic variants that affect how drugs work in your body. And you can see that more people, 85%, have at least one actionable variant, but more people have more than one actionable variant that would affect how your body can process and use and respond to one of 50 common medications. And then on the right panel is a report from a new clinical DNA test that has been developed and released and is available to any patient in the country now. Providers can order this result. It's a polygenic risk score, which takes millions of genetic variants and aggregates their differences to produce risk scores. And this is what the summary looks like. So it tells you whether you're below average, average, above average, or high for one of 8 different cardiovascular conditions. These genetic conditions are ones that we can act on, and you can discuss with your provider. And it importantly was trained on all of us and then validated in a separate population. I want to dig just a little bit deeper into what it looks like to be one of those 5,600 patients that have received potentially life-saving results. And here's just 3 examples of stories of people in the program. The first is a participant in his 60s that had no relevant family history, who learned of a pathogenic variant in the RET gene, which is associated with multiple endocrine neoplasia type 2. This individual was later worked up clinically. Thyroid ultrasound revealed a 1.5 centimeter mass, which biopsy determined to be medullary thyroid carcinoma. Notably, his calcitonin was over 1,000, which is incredibly elevated. He had what seemed to be a curative thyroidectomy and lymph node dissection. And notably, his sibling also has the same variant who is currently undergoing surveillance. Another example is a 29-year-old who joined all of us to contribute to science, and he learned of a KCNQ1 mutation, which makes him susceptible to long QT syndrome. Following testing with a cardiologist, this individual started taking a beta blocker to reduce his risk of sudden cardiac death from long QT. And then finally, a 35-year-old who opted in to receive results. 35-year-olds are not recommended to get routine breast cancer screening. This individual is found to have a pathogenic BRCA2 variant, which puts individuals at increased risk of cancer, notably breast and ovarian cancer. With this information, she started earlier screening, including MRIs, and was found to have an early stage breast cancer, which at this point is highly treatable. But at later points with less screening, interventions and intensive screening may not have been. So these are dramatic changes in these people and represent just 3 of 5,600 participants that have received these kinds of results and potentially found intervening conditions as a result of our program. Now, as we do this, of course, our goal has been to accelerate research. Returning results is a byproduct of the work that we're doing and an important focus of what we tried to do. As we have interpreted those genetic variants across so many different individuals, we have returned and processed and called and interpreted more than 40,000 genetic variants, and 8,000 have not been classified before. This puts us in the top 1% of all clinical labs that contribute to ClinVar, which is a resource that providers use to interpret genetic variants. We're not a clinical lab, but we follow those procedures and are providing important data to important genes that other clinicians can use. And these data accelerate discovery in many different ways. We'll kind of talk through a couple examples. These are just a couple of them. One of the ones we have here, I'll just highlight because we don't have other examples in this deck, about step counts and the importance of the step counts to impacting chronic disease in this example. And also, there's another one referencing using AI to help and validate potential repurposed drugs that might help with Alzheimer's and other dementias. We want to take our publications and make them understandable for participants as well, and also just for researchers to quickly understand what's happening. We used to do this manually, and that has become fairly untenable. And so over the course of 2025, we developed a large language model pipeline that is fed by automatic ingestion right now from PubMed. We will be expanding that to other resources as well. And we use this to classify whether a publication is actually using all of us data, and also what type of publication. Is it genetic studies? Look at chronic disease, look at rare disease, things like that. And those are available with filters. But more recently, we've also developed a automatic lay language translation system, which you can see there for this example study that is looking at genetic variants in coenzyme Q10 and the issue and potential for having statin-associated muscle symptoms, and use All of Us and the UK Biobank. And this study, for instance, you can read the description there. It just describes that people that had genetic variants in coenzyme Q10 were actually more likely to have muscle side effects when using statins. And it talks about it at, I think, a fairly high level, and hopefully more interpretable for a participant. And what's important about this study, just to highlight, is many people do take coenzyme Q10 when they're on statins, and there is evidence to suggest that that does help with statin-based muscle symptoms. And this study shows that genetic variants are, as a surrogate for that, provide evidence for that potentially being a helpful item for people who take statins and have those side effects. So it's providing genetic evidence for treatment. Our genomic data was one of the hallmarks of what we've been doing in this program, and about 50% of our participants for whom we have genomic data come from individuals who are most closely similar to non-European genetic population. This is important because more than 90% of all genetic cohorts out there in genetic studies have represented people that are most similar to European genetic ancestry, and that results in us having a harder time interpreting genetic variants, calling pathogenicity, making polygenic risk scores that are applicable to everyone. And this just shows an example survey and study with looking at lipids and a number of significant results. That this study replicated associations with over 117 diseases, showing that it is broadly efficient for doing gene disease discovery, and also talked about just the number of novel genetic variants we have found. Over 275 million genetic variants. In this early study, this is on an early release of our data, over 275 million genetic variants were previously unreported. To put that in context, many of the consumer genetic tests would only look at a million genetic variants. When you're looking at ancestry, we have about 3 billion base pairs, and this just having this large number of genetic variants helps us start to understand what the impact of this rare variation is. Some of it will cause disease, much of it will do nothing, and is just part of normal human variation without a health impact. And knowing the difference is hard. You have to have the references, populations to help us understand over time what the difference is. And then to dive in a little bit more, chronic kidney disease, of course, is a major cause of morbidity and mortality. There's about a 50% more 5-year mortality of individuals once they enter end-stage kidney disease. So huge impact on public health and obviously a huge cost to the system. African Americans have about a 4-fold increased risk of developing end-stage chronic kidney disease, and most of that is due to that excess risk is due to APOL1. Individuals with 2 genetic variants for APOL1 have about a 70% excess risk of end-stage kidney disease. And in this study, using 3 cohorts, it's found that this rare genetic variant, N264K, which is in the APOL1 channel, actually alleviates and lessens that risk of having end-stage kidney disease. This is important because it shows a mechanism of action for hopefully preventing APOL1 kidney disease. And importantly, there are several drugs that are in clinical trials that operate through this exact mechanism. I'll highlight here in this example, all of us is not the tallest bar in the study, but the current data release that's coming out soon will actually be 5 times as large as the data set that was used in the study. And currently, there are over 70 projects in all of us that are looking at this very condition to help advance science in this space. I talked about polygenic risk scores and the clinical tests on the market, and here's another study that used all of us to take polygenic risk scores, make them relevant for diverse populations so that they could be implemented in a clinical trial, in this case, NHGRI's funded eMERGE network, to look at 25,000 individuals in a randomized format to be able to get polygenic risk scores, as well as clinical factors to look at the impact that will have on disease risk. And so you can see this here. You can see the number of individuals, for instance, that have a high PRS risk threshold. What's really key here is, you can see the odds ratios now generally work across all relevant populations that are going to be in the study. And without using all of us, those effect sizes, those effect estimates would not have been as nearly as accurate to deploy in this clinical trial. Here's another study. Epstein-Barr virus is a common virus, causes infectious mononucleosis. Most of us are exposed to it sometime before we're 20. And in this study, they used genetic data to find evidence of having had Epstein-Barr virus and use that to look at the disease impacts. They found a population 100 times larger than previous research using Epstein-Barr virus by looking at sequence evidence. The sequence evidence of having persistent Epstein-Barr virus was much more predictive of disease and in a much larger population than what researchers found by using typical antibody testing. And you can see that there's a whole host of conditions here that are associated with having DNA circulating evidence of having Epstein-Barr virus there. And this suggests more of our autoimmune disease and some other conditions may be related to this. Here's another study looking at prostate cancer. In this case, another polygenic risk score was trained on prostate cancer. On the right, you see the risk of having prostate cancer or metastatic prostate cancer in B, given by different percentiles on the polygenic risk score. After being trained in all of us and other systems, this polygenic risk score is being implemented in a randomized control trial with 5,000 veterans in the VA hospital system. And so this will be a powerful study, we believe, to help us understand the role of polygenic risk scores to add to clinical care in a real world system with a real disease that is common and quite variable in its impact, but yet significantly causes morbidity and mortality in a subset of the individuals that get prostate cancer. And hopefully this will yield positive results there and guide our thinking in the future. Everyone's talking about AI in many ways now, and I just wanted to highlight one study that trained in a safe way a large language model within our research ecosystem to look at the prediction of disease risk using clinical factors from the electronic health record as well as polygenic risk scores. And this is a high level, simple way to aggregate genetic changes in this risk. But you can see here that in blue, using just EHR data, red, using EHR data with polygenic risk scores, that the team saw improvement across these four conditions: type 2 diabetes, schizophrenia, depression, and coronary artery disease, and in general improved by incorporating at a high level genetic information. And it's the first step to many such studies, but it just shows the ability to do this kind of work within our workbench, and I want to highlight. Wearable data is an important kind of data for helping us predict activity. We know activity is good, and we have longitudinal wearable data currently available on 59,000 participants with more than 39 million days of step counts and 31 million nights of sleep. Having this longitudinal information is so important for thinking about prediction of many things, and researchers are using this for innovative ways to look at recovery from surgery, changes after pregnancy, how treatments in surgery and post-surgery with pain medications affect things like recovery and complications and sleep quality. And so we started in black with a bring your own device program. In blue, we added what we call our WEAR program, which gave devices to people to diversify the population and create a greater capture of individuals who have these kinds of devices in a longitudinal way to think about disease risk and health. We want to make tools that support research really for everyone. And so and these are just some examples of some of the tools. We have recently calculated an all genotypes by all phenotypes, so that researchers can quickly get a sense of whether there might be associations in these data. And this was over 200 years worth of compute, and it makes it available quickly for researchers to find this. And you can explore that with the URL there. We have genomic variant browsers. We have an imputation service that we have developed with NHGRI and NHGRI support to help develop this using Anvil data as well as All of Us data, the vast majority of it being from All of Us. And so I just want to point out on this panel on the left, these are a number of different imputation servers that researchers can use. And one of the most common at this point would be TopMed, which has done a great job representing different genetic ancestries to calculate haplotypes and impute well. But you can see our non-European genetic ancestry population would already be the biggest imputation server in the world. And so overall, it's been trained with 515,000 individuals. And you can see that improves imputation quality on the graphs on the right. Compared to TopMed across every genetic ancestry so far that we have compared. We want to make data and science reproducible, fast, and onboard as many researchers as possible. And so we have lots of public tools. We also have tools that, of course, are only available to register researchers. And you go through that step, that's 1 through 6 of confirming institutional access and verifying identity, completing the code of conduct, and then starting to do research in here. And we find that researchers can get started in a median of 29 hours from when they first log into the website. And there's no IRB approval needed for this kind of research. It is non-human subject research in this protected cloud model. And in fact, you even get $300 of credit to start. And so many of us are used to the paradigm on top of a traditional research process taking years to start. Other cohorts can take 9 to 12 months to get access, and I've used many different cohorts, and many of you probably have where you go through and get a project approval, write that up, and then get approval for just that project and start doing work. In this process, if you're one of the 1,300 institutions that already has a master agreement with us, we can validate your identity, we can validate your affiliation with the institution, and let you write up that description, review it asynchronously with our resource access board and automated approaches as needed. And you can get started doing real work in as little as 2 hours from when you first log in. This has, I think, been one of the key efforts to helping us grow our research population base. And you can see that we're now over 20,000 researchers. The publications are growing non-linearly, and we're really grateful to see that and should really be taking off. And at the bottom, I just highlight the number of really important engagement partners we've had on the researcher side, engaging researchers across different parts of this country in different ways and in different carrying the message forward, helping them on board. And as we talk about the budget cut to come, unfortunately, we won't be able to keep the full cadre of researcher support that we have had before. But we are very grateful that they've helped us get here. This resource, as it exists now, is a resource that has being used by a number of researchers that are not affiliated with all of us, writing research project grants, now over 300, to use the program. And you can see this is growing each year. Total, we've had over $600 million of research grants that get to use our data for free. And so, just think if they had to enroll those cohorts, how much more that would have cost in that process. And you can see it's supporting in early stage investigators, new investigators, and indeed we have a number of intramural NIH researchers, and it really does cover across all institutes and centers, both extramurally and intramurally. This gives you a sense of our data releases, and dark blue is what's currently available, and light blue is what's coming in June. So towards the end of June, we will release a data set that will have over 750,000 people with survey responses, or 535,000 with short read whole genomes. And most all of those will have electronic health record data. There will be more people with wearable data, more people with other types of data. And importantly, this is really our first multi-omic data set. We'll have 14,000 with long read genomes, including methylation data. Most of those 14,000, 10,000 of them will also have proteomic data. Many will also have transcriptomic data. We'll later be releasing metabolomic data. We've done our first foray into extracts from narrative text from clinical notes, and that will represent 90 million notes we'll have extracts from as a pilot. We'll be bringing on over time Apple Health Kit data and more environmental data through geospatial linkages. We will also be starting, and actually already have started, to a number of tens of thousands of people actually, our first reassessment, where we ask people to take some of the initial studies and surveys that they did at the beginning. This is also accompanied with a $10 gift card, and it updates and adds to some of the studies and surveys we do when people enroll. We mentioned earlier the launch in pediatrics, which has been running in a small way because of our budget cuts, but we still have been able to find ways to expand this and actually grow more fastly. And you can see with over 3,000 now, we collect, importantly, similar types of data as we do on the adult side. All current pediatric participants have a parent or guardian who's also enrolled in the program. And then when we get to teenagers, that will be optional. This year will be expanding to ages 10 and younger. So right now it's 0 to 4. And we're also going to add a way that this can be done nationally through virtual enrollment. So any parent or guardian that's currently an All of Us participant will be able to enroll their children later this year. And even though we have a small number of kids that have enrolled primarily, we have a lot of extant data that are shared from electronic health records of people when they used to be kids. So in this case, for 40,000 people who have shared pediatric data, it'll be over 60,000 in the last release. In the next release. And over 55,000 in the next release have whole genome sequence data and pediatric data. We did our first Science Day on April 15th. We had over 2,000 people attend live, and then over 500 that have watched views since then. And then we had lots of other kinds of sessions here. We had attendees that were both participants and researchers. And so we really tried to do both. And so one of these quotes that I loved here, the data I contribute today will outlive me, eventually shared by a researcher not yet born, using technology not yet imagined at a conference not yet planned. And this was from a participant who was attending Science Day for us. We have engaged participants through other engagement forums with over 5,000 participants engaged. These are typically 90-minute forums, which provide some updates, do live Q&A. So we have to keep the number small enough that we can support that live Q&A. And we do different regions. And we have another one coming up on June 9th. Encourage any participants watching. If you want to join, you'll see information coming on this with future ones on reassessment and veterans health. Our system is a platform for partnered research studies, we call them. People launch new studies on top of us. So with the partner research studies, we have more than 20 in the pipeline at various stages. 2 have been completed, 7 are ongoing. The largest of these is Nutrition for Precision Health. It is a large study that's deeply enrolling 8,000 people that share data about diet, you have microbiome assessment, metabolomic assessment, and a number of different other kinds of information. We have exposomic information that's coming in another study called Environmental Health and Exposomics, that's looking at people who develop type 2 diabetes after they enrolled in programs. So we'll be able to look at gene by environment risk factors for incident diabetes. Eyes on Health is doing eye imaging on 5,000 participants in 4 different sites, and a number of other ones you can read here. One that I'll just highlight starting soon, this summer will be Genotype to Phenotype, which is a program that will actually bring people into the NIH Clinical Center to do deep phenotyping based on the presence of rare genetic variants in either known conditions or potentially unknown conditions that we think might be genetic conditions, to understand and obviously provide information back to those participants as well. Really excited about what's coming with external investigator access. And this just happened yesterday. I'm excited to announce that our EXO-1 that you may have heard us talk about in other forums is now out on the street and is open for applications, accelerating discovery through partnered research with the All of Us program to analyze participant biosamples. And so this will give outside researchers a chance to propose ideas to use our banked biospecimens, plasma, serum, and DNA, to generate new types of data. And so an EXO-1 does not provide funding, but it does provide that competitive access. And you can use funding from any number of other kinds of resources, whether it be academic funding, grants, for-profit, to propose ideas to generate these data. All the data that are generated will be returned to the researcher workbench for other people to use as well. And it obviously gives this kind of opportunity for engaging all of us participants. We ask everyone who does this to think about what kinds of value they can return as well, whether that be a educational document or something else, and we will be guiding more about this. This happened really fast, and so in terms of it actually becoming live, and so we're actually still making our links live, so stay tuned. You will see more information coming from us on this and other program announcements to explain more. So finally, I want to end with just a story of where we are in this program and how we're thinking about the future. This graph shows in blue the funding that we have received from base funding, and then in orange, the other source of funding we received from the 21st Century Cures Act, which ends with fiscal year 26. Because of the changes that happened in the expansion of 21st Century Cures in fiscal year 23, you could see our total had stayed the same, but our base decreased, and it has not changed since. We, in fiscal year 27, will be at a level that's 8 million less than when, before there was a program. This requires us to think very soberly about what's happened in the program. And people that are with the program, certainly our awardees have experienced a number of cuts in the last couple of years in understanding. This next level will be hard. We will firmly commit to protecting our participant data, our participant ability to access the platform, and our biosamples. Data security is not being sacrificed. Those are things that we have to focus on and will continue to support. However, And many of other things will receive significant cuts, and things like returning genetic results like we've done powerfully before. I'm not sure how we're going to support in the future. We will continue to be innovative and see what we can do in these spaces, but right now we don't have the funding to do that. Our network of healthcare provider organizations has seen and will see dramatic changes. And certainly, I already mentioned things like researcher support and other things. And so we will keep researcher access, we will keep the matriculation, we will not be able to support the enrollment like we had before. Clearly, we would have had over 1 million participants by now if the funding level had continued as it was. As it is, we're grateful that we have an incredibly important and large and powerful data set that our participants have donated into the system. And so with that, I'll just say thank you most of all to our participants. Thank you for staying with us and staying a little bit long for this open session. And I'm happy to take any questions as we have time.
Thank you so much, Josh. And yes, for everyone who's able to stay for just a couple more minutes, we're happy to share some of the questions that we received both in advance as well as during this session. So just to be clear, Josh, will all of us continue to recruit new participants?
We will continue to be open to new participants joining digitally. Where we don't have funding is to support the active enrollment of new participants, collection of biosamples, and really are a way we've engaged and really sought to form those relationships. Most of the enrollment that will happen in the future will come through partnered research studies. So as people partner with us, we are trying to maintain, and even this is hard to be honest, we are trying to maintain the ability to allow partners to use us to recruit as they provide funding to do so.
Thank you. Related to sort of priority enrollment or partnered research studies, how has All of Us addressed the opioid epidemic?
We had a partnership with NIDA, National Institute for Drug Abuse, to focus on enrollment on individuals that are at risk for substance use disorders. And so they partnered, this is an example of a partner research study that was focused on enrollment. They worked with another number of sites to enroll, and that was actually something we were able to spin up actually in months and enroll, actually exceed the goals that the program had had. So it was an efficient process and effectively represented those populations. In addition, NIDA did a data challenge on our program and had the second most respondents ever for one of their data challenges. And they presented a number of innovative proposals, and people have been looking at things like GLP-1s for substance abuse, genetics, both pharmacogenetics and polygenic risk scores. There's a number of research projects you see that are being generated on our program to support understanding and alleviation of substance use disorders.
So, for example, as some of that data is, put into the pipeline from more recent priority enrollment, and then we're looking at future researcher workbench data releases, you reviewed the summertime next big batch of data that's coming out. Can you talk a little bit about the longer term future of the researcher workbench and whether they're on cycle or off cycle data releases, like what could be coming?
In June, we will have our next version of the data set that will come out as a large data set. As we move into the future, and even through 2026 and 27, you'll see a number of what we call off-cycle data releases that will be less about maybe one large data set and more about a large data set and then other subsequent updates that happen as these off-cycle data sets. So some of the multi-omic data I talked about will come as off-cycle data releases, mostly in 2026, but maybe some in 27.
Thank you. I'll do one more question and then we'll end. You may have seen recently in the news, PCOS, polycystic ovarian syndrome, was recently renamed to PMOS. How would something like that in the future be sort of incorporated, as diseases get updated and syndromes and so forth?
This has been replete. I even remembered hepatitis C being called non-A, non-B hepatitis. And certainly I'm not that old that I was primarily with that, but you'd see that in the record of patients I used to see. And so, these kinds of changes happen over time. Fortunately, we take all the data that we get from electronic health records and our surveys and things into a common data model that supports the evolution of these terminologies over time. And so as these changes happen, in some cases, the verbiage you'll see in the site may lag a little bit. What is the current standards? But importantly, the data usually are mapped well, and we keep an eye out for that sort of thing.
Thanks, Josh. As we think about the future of all of us, we have different comments that I'm seeing here of people, giving their support, but what are some maybe more tangible ways that participants or researchers or the public could show their support for the program?
Well, first of all, I thank everyone again who is a participant for joining. I thank all the researchers that are part of it because the data has to be used to make a difference. And so, the first thing is I always think, if you're a participant, stay engaged. We have a new participant platform. If you haven't logged in the last year or so, come back. And there's probably something new that you may have to do. And so that is always the first thing I think about. The next thing though is, if you feel strongly and want to reach out to your congressperson, for support of the program, feel free. If you have other means or advocacy, that's certainly we would appreciate that kind of support. Op-eds, things like that as well.
Thank you so much, Josh. So with that, we'll wrap it up. And if you want to say anything in closing, but we really appreciate everybody who joined us today, and we will be putting a recording of this up on our YouTube channel.
Thank you all.